And my intent was to start with the acknowledgments up front in case I don't get there later, and that is to say that this is sort of an integrated effort from part of the Rock Powered Life team, and we thank the NASA Astrobiology Institute for funding. And my goal today is going to be to integrate data that's come from several different students that have been working on various aspects of the system. So you'll see me refer to them, but this is taking just some of the snapshots of current information from a variety of different studies looking at both the geochemistry and integrated microbiology derived from each of these students and postdocs right now. So the system that I'm going to talk about today is derived from just one of the sites that we work on in RPL. I'm specifically going to look at the Oman ophiolite, and the reason here is that I'm trying to look into the subsurface of a system where we have an enormous volumetric amount of peridotite that's not dramatically impacted by other lithologies surrounding it. And so what we're able to do is to look into a subsurface environment where the peridotite is an aquifer that's active, it's storing fluids, and those fluids are in the 30 to 50 degrees C range, and they have abundant hydrogen. So I'm not going to be talking about the generation of that hydrogen today, but rather to say everywhere we look, we have anywhere from micromolar to millimolar concentrations of hydrogen, and that is being modulated again by not only the geochemical dynamics, but the microbial activity within the subsurface. And in terms of linking this to some of the systems that I think this information is going to be relevant for, I wanted to put up both of these slides today. So when we talk about looking at subsurface life in a peridotite in, let's say, an icy worlds context, most of the time, at least for what we're working on in the Oman system, we're not discussing really the ocean and the possible biological activity in the ocean system or at the under surface of the ice, although serpentinization, the process we're studying, is well known to modulate potentially the ocean chemistry. Rather, we're interested in imagining what may occur in the rock-hosted environment and what those habitable niches are as you have fluid ingress and circulation at temperatures that are non-hydrothermal. So we're looking at what would be represented by large volumes of rock again, but away from a high heat source. When we talk about Mars, which is something that I referred to really briefly in part of the plenary yesterday, in this particular case, we're interested in our system in the more static component of the subsurface environment, and I'll differentiate that today. And these are areas where we're really not having aggressive fluid circulation or exchange. It's a relatively sealed system, but we would like to know if you had a subsurface aquifer on Mars and you had hydrogen stored or generated in that condition, what is it required to allow biological activity to persist over long periods of time? And if that should happen, what would be both the signals of that extant life, or again, if it was to be preserved or fossilized, what would we expect to see as those signals? So contextually, again, I just keep coming back to where water-rock interaction, but under highly rock-dominated conditions, but trying to understand the microbial activity and preservation under that state. So if I'm lucky, now it's maybe almost about 2:30 and I can just talk about data. Okay, so Oman system, cartoon format. When I show this type of a picture of the surface environment of peridotite, that would sort of be here, looking at a mountain landscape. What we're interested in is looking then again at the hydrology in the rock system in the deeper subsurface. And we've been really fortunate to have access to the subsurface through pre-existing legacy boreholes, and that's a way that we're able to pull fluids out of the subsurface over a diverse array of conditions, as well as targeted drilling in the last year, in 2018, that has allowed us to pull and recover core over many hundreds of meters in multiple locations. And those boreholes, or again, the cord rocks, they're penetrating different parts of a hydrological regime. So there's a shallow flow system, shorter residence time fluids moving through a fracture network in habitable conditions generated here. And James Long gave a great talk earlier this week about some of the geochemical states of that system. But we're also really interested in the ability of life to persist and thrive and be active and preserved in the deeper parts here, which I'm going to call the more closed system. It's more hyperalkaline and highly reducing. From having pulled a lot of fluids out year after year and across the different parts of the system, we do know a lot about who the dominant organisms at least are biologically. So Katie Rumford published a paper, and following on from Hannah Miller, really identifying, for example, acetothermia, thermatosulfovibrio, methanobacterium, several different organisms as some of the dominant, again, methane, acetogenic, sulfur cycling, and nitrogen cycling taxa that are present. Now, they're differentially distributed, and we're still trying to probe then the activity of different organisms in different states of the system to understand their role. If we take one environment where we're really profiling as a function of depth and we look at the different geochemical states of the system, this is where we're in a suboxic, moderately reducing environment that we can find often in the shallower parts, the upper tens of meters in the fractured aquifer. But as soon as we get into a part of the system that, again, has a lot less fluid transfer that's occurring through, it's more static and closed, we rapidly drive down to the lower stability of limit of water and are held there. And so we actually have a huge amount of data that sits right down here, but we can span these conditions. When we pump fluids, sorry about that, and when we pump fluids, we're pumping all of this up together and integrating it into that community. So a lot of our goals at the moment have been to actually, as a function of depth and in geochemical environments, to separate this out better. But if we're in the surface environment where there's a lot of fluid mixing going on, nitrate is our most dominant electron acceptor. And so Katie Remfert's been trying to trace the prevalence of nitrate, and then we have full conversion to ammonia. So this becomes a highly reducing system, high ammonia fluids, and to look at the differential gene abundance for nitrogen cycling. The idea here would be, although we have nitrate in some of these sort of more shallow mixing systems, it's really not playing a role deeper down. We do have sulfate throughout. We often have sulfate in excess that's not being fully consumed. We do lose our DIC, but we do have partially carbonated rocks. And each of these set a state of the system that's been interesting to probe biologically. In some work published this year by Libby Phones, one of the things that we're able to see is that no matter where we've been pulling fluids from in this more integrated fashion, we're often sitting at about 10 to the 5 cells per mil. This is actually a surprisingly robust microbial population that we're seeing in the fluid system throughout. And it's true even when we're in these more hyperalkaline fluids, meaning once we're above pH of 10. And in the same study, one of the things that Libby was working on was trying to probe both the reduction of CO2 through a variety of different pathways and its assimilation into biomass. In this particular assay, what she was doing was trying to take 14 labeled bicarbonate and look at its conversion to methane to detect active methanogenesis. Again, what she can see is that there's active methanogenesis through all the different pH and water chemistries of the system, although, I'll leave it there for right now. So this was promising to us. We also see methanogens distributed throughout the system. However, when we come in and we start to look at the geochemistry of the fluids and just the methane alone, it's often quite confounding in terms of how to interpret the data. So this is some work from Dan Notaft. And in this particular case, what he's plotting is the delta 13C of methane against C1 over C2 plus C3 alkanes. In our surface fractured, shallow mixed water kind of aquifers, we get pulses of methane out, and they're easily recognized as microbial. We also get these higher rates of microbial methanogenesis there. There's really no question. However, when we move to the closed system hyperalkaline part, we have this enormous flip in terms of the delta 13C of methane. We're often sitting right around 0 per mil. And it's very enigmatic to interpret it in terms of its origin and source. But one of the things that's notable is when you look at C1 over the C2 plus C3, we're 10 to the 2 to 10 to the 5 in terms of enrichment. And it again points to this potential for a microbial source, but we need to have other ways to assess and discriminate that. And it would be surprising. It'd be surprising to have methane with compositions as heavy as this that may be generated through biological activity, particularly because this is the state of the fluids also in which we're looking and trying to probe the activity of methanogens. Sitting at the lower stability limit of water, sitting across these alkaline to hyperalkaline pH and very limited DIC. But these are in rocks that have undergone significant water-rock reaction and are carbonated, even though it's a low level of carbonation throughout. So coming back to the geochemical parameters, one of the things that Dan did is he worked with both Shuhei Ono's lab and then with Ed Young's lab, trying to look at the clumped isotope systematics of the methane. When we started this project, the only parameter available to us here was looking at the CAP13CH3D component. And we're just looking at this case at different systems at what would be an isotopic equilibrium. And in this case, we have, again, significant bond disorder and a partitioning of methane here that looks like it's microbial in nature. This year, we were able to get an extra lens into this through the CAP12C H2D2, as well as in this kinds of systems. And in this case, this would be an array for both low to high temperature isotopic equilibrium. But when we're looking at these two rare mass 18 isotopologues, this would be, for example, data from Kidd Creek. But this is the microbial array, and Aman's plotting squarely within that as well from this lens. So coming back to then trying to probe these systems in terms of the active biology, Emily Krauss gave a talk here earlier this week on Tuesday, where she's going in and pulling DNA again out of these highly reduced hyperalkaline fluids. And in sequencing in this particular set of samples that we've had recently, remember again, I had 10 to the 5 cells per mil as an average number across the system. And now we have 10 to almost 20% of the total 16 S reads are from methanobacterium that's present. That's a pretty abundant methanogenic population in these hyperalkaline reduced fluids. And then in this other axis here, what she's looking at is the number of transcripts. And so the methanogens are by far the active population under this condition. So we are getting a number of metrics that are telling us they're here and they're busy and they are making methane, and we're capturing and collecting that with a pretty strong signal from both the geochemical and biological space. We have been very fortunate working here that we had done targeted culturing that started a few years ago, going after methanogenesis. And we do have one of the methanobacterium in culture that's present in the system. So Dan Coleman's actually mapping out the differential distribution of methanobacterium across the system. This is one of the methanobacterium that we see. And Hannah Miller was able to then start to grow it in the laboratory, where she was providing it with a variety of carbon sources, including trying to grow it on calcium carbonate. And the idea here is with this particular isolate, she's able to grow it much more easily in these more alkaline pH, high DIC conditions, but she can push it to higher pH and slower growth rates, but still get persistent methane formation and see a huge shift in the delta 13C of methane that increases by over 50 per mil. And that's just a simple function of carbon limitation and availability in that system, but it's a strong switch that's flipping us in terms of what that bulk isotope characteristic is. So to give an image of that, what I'm envisioning is that back in the field site type of system is that when we're looking in areas where there's been this prior carbonation, even though there's very little DIC and we're sitting in hyperalkaline pH, the rate and solubilization of calcium carbonate is very low. But we must have a coupled acquisition system here, which is allowing there to be the solubilization, uptake, and almost quantitative conversion of that CO2 into methane. And that's something that we're now trying to probe and get at the rates for. And listening to Sanjoy's talk earlier today, because it is fun to try and integrate through the week, it's been really interesting to have these discussions about the KM for methanogenesis for this step, for CO2 acquisition, and to think about whether or not this organism or some of the ones that are adapted in here maybe do have a different KM that affects some of the energetics. And that's something that hasn't been directly tested or probed. So just the last part of this talk, I'm just have a slide or two to also connect to some of the work we've been doing looking at sulfate reduction. So this is with Clemens Golombitsa, who is an NPP postdoc in Tori Holler's lab at NASA Ames. And this is another incredibly astrobiologically relevant metabolism for these systems, looking at sulfate as the dominant electron acceptor, not only with hydrogen, but also with the micromolar concentrations of organic acids we detect in these systems, as well as with the abundant methane. And so Clemens systematically started probing the fluid system to start with a 35 labeled sulfate to try and probe the distribution of sulfate reduction activity. And in general, he has very low rates, so picomoles per mils per day. He's worked in deep sea sediments in different parts of the globe, and these are exceedingly low rates, but he has been able to see them persistent, but they are really diminished in the hyperalkaline state of the system. So unlike methanogenesis, sulfate reduction is actually the one hitting up against a wall in ways we don't understand once we get above pH of 10. Now that we've done the drilling and and we are starting to look as a function of depth and again, geochemical states of the system and the subsurface, Clemens has again been probing these cores to look at the differential distribution of sulfate reduction activity. So in just one of the cores that we've been starting to work on, and this would go from 0 to 400 meters of depth, or at least this core does, he probed a series of different depths and has a clear localization of where sulfate reduction is occurring. We have downhole logs that allow us to look at the pH and the redox EH of these fluids. And what we're really finding is that once that's a reducing condition, but we drop down below pH 10 or 10.5, we see the sulfate reduction kick in, and it's being suppressed elsewhere in those cores. So this is giving us some guideposts now and other work that's ongoing, such as Katie Remford's talk earlier this week, where she's been looking at the intact polar lipid distribution in these samples. This has been a great place for her to work to prove that she can really extract and start to characterize IPLs from these kinds of cores and detect them well in excess of any contamination in these kinds of systems. So to conclude, we're at the beginning, and we're really feeling like we're now starting to understand components of how the system is set up, that we can begin to really aggressively delineate where different microbial metabolisms are functionally active and start working on pulling out and exploring the physiological adaptations to hyperalkaline pH and this extreme carbon limitation and the differential abundance and activity of populations. We are very interested in both the total community but the active microbial community in these very different parts of the system, that more open, mixed chemical environment, and then what's sealed into the more static aspect of the system but is able to slowly persist. And all of those, of course, are going to lend themselves to trying to identify the biomarkers and the biosignatures of that activity. We have to keep nesting in at these different scales to come find that, but our search strategy for life and actually trying to represent the chemical and the biological measures of that life activity is looking really promising. So thank you very much.
Thanks, Alexis. We have time for a few questions for Alexis.
You mentioned Kid Creek and the clumped isotopes, and then with the sulfate reduction, you also show that methane goes to bicarbonate. Is there a carbon cycling there that, I mean, how does that fit into the, I forget exactly what you said in reference to the where your data fit into the Kid Creek, but are there methanotropes, you know, coupled to sulfur that affect the clump isotope volume?
Yeah, I mean, it's in the overall question of what you're asking. We have a really complex carbon cycle occurring here, and we're just feeling like we're starting to have the right data to tease it out. So, especially in places where there's no measurable DIC, I mean, there's a lot of questions about what's the real form of carbon being taken up by cells and being used. So, CO2 is one option, and we have this discussion about the KM, but we're really looking at formate and CO availability and its role in potentially, for example, with the methanogenesis and the sulfate reducing populations. But we have to start to really trace that elegantly, and we've had to know where and how to do that. So I think we're at the tip of the iceberg and trying to really understand the balance of how we're both producing and reconsuming CO2 and where these different populations are active. It's weird that we can shut the sulfate reduction down above pH 10 and really not be able to detect it. That allows us to separate sulfate reduction and the methanogen activity at the higher pH conditions and re-put them back together in the moderate pH 10, 9 kind of realm.
We have time for another question for Alexis, if there are others.
Thanks for the great talk. I have a related question, but maybe it's too specific, but I'm curious about it. If you have any insights as to the membrane bioenergetics of these organisms, because one of the persistent questions is the PMF, or is it a sodium motive force, or do you guys have any new data about that?
We don't have new data about that. What we are doing is actually using data from some of the work Dan Coleman's been doing, which isn't shown here, trying to come back in and actually retarget the isolation of the methanobacterium that is functioning well in the hyperalkaline component of the system, so the one that's really differentially distributed there. And that one would be a more appropriate one to do this. I'm also really interested generally with the methanogens, what the delta pH is between the external and the internal environment. And I think we really need a good measure of what is that intracellular pH, which is also part of then these membrane bioenergetics. So we're thinking about that, but how to do this well. And we really would like to move beyond the methanobacterium we have in culture and go after the one that we think is more physiologically adapted.
Great, thank you, Alexis.